So third generation sequencing, also called TGS short form, DNA sequencing, it's technology that's allow directly sequencing. We don't need any labor work, not any hectic rules for that. We get DNA fragment, provide significant advantages in genomic and molecular biology. After this third generation sequencing, we have a lot of significant advancement in the genomics and molecular. Overview of third generation sequencing. This sequencing technique is the advancement in the DNA sequencing technology, developed primarily in 2010. It's not like the previous generation sequencing like first generation and second generation, which typically produce short read. This generation sequencing, third generation sequencing, is used and can use for the long read. It's often exceed 10,000 base pairs and even much more, several million base pair in some cases. Third generation sequencing have capability as crucial for accuracy, assembling genome and studying complex region. Key techniques in third generation, we have two key techniques: small molecule sequencing in real time that's developed by Pacific Biosciences. This method allows us to use real-time observation of DNA analysis, enabling the sequencing of single molecule. We don't need the amplification process. Export Nanopore. This technique involves passing DNA molecule through nanopore and measuring the change in the electrical current to determine the sequence of base. I have a technique that measures the change of electrical current. On the basis of changes in the current, we identify sequence of base, either it's adenine, cytosine, thymine, or guanine. It is notable for its probability and ability to sequence DNA outside traditional lab setting. We can use and pick in the pocket. We can sequence DNA outside of the lab. What are the advantages of third generation sequencing? First of all, long reads. The second one is real-time sequencing, and third one is single molecule sequencing. Long read sequence are the studies of DNA which help in the resolving repetitive region and complex genomic structure, and that are the challenges for the short read technology. Short read technology was only for dealing with the short read, but third generation sequencing are dealing with long read. Real-time sequencing, this is the ability of third generation sequencing through which we analyze DNA in real time. It allows immediately data acquisition and processing, which can accelerate research timeline. Moreover, single molecule sequencing, third generation sequence eliminates the need of polymerase chain reaction amplification. It's reduced biases and also allowing for a more accurate representation of the original nucleic acid sample. Application of third generation sequencing or genomic assembly, structure variation detection, either it's SNPs, CNVs, and clinical diagnosis. So this is the overall graphical visualization and one picture. First generation shotgun sequencing, second generation massively parallel sequencing, third generation single molecule sequencing. So we are clearly know how they are changes from one and other. And first generation is not more advanced than second. Second is not more advanced than third. So it was the advancement in the sequencing that was very helpful, and also it is very helpful still for research and molecular biology. So overall sequence species, from where what was the first species, either it was prokaryotic or it was eukaryotic organism that was sequenced. In 1965, first yeast cell was sequenced. The size was 76 base pair. After that, bacteriophage virus become to be sequenced and the size was 12 base pair. And after that, Escherichia coli RNA was sequenced and the size was 120 base pair. So on and so forth. In 1990, Human Genome Project sequencing start, and also it's completed in 2003 and the size was 2.9 base GBs. And after that, mouse, dog, rat, chimpanzee, so on and so forth, eukaryotic organism started to sequencing. And after that, genomic and VGP, every research field and also some marine animals like zebrafish become sequenced up to 2023. These are the list from where we start the sequencing. What was the first organism that become sequenced sequence, and it was yeast and bacterial phage. So NGS complete workflow how it's performed. So first in laboratory we perform experimental work to extract DNA. After that sample preparation and library generation. It's RNA extraction. Okay. After that polymerase reaction start with the addition of polyethylene, mRNA purification. After that perform mRNA fragmentation. After that start reverse transcriptase to become cDNA, complementary DNA. Adapter was added. After that clonical amplification PCR performed. After that high throughput sequencing performed. We get raw data. After getting raw data, we can perform various bioinformatics techniques so that we provide biological insight to the data. And after that, bioinformatics has started working to get and analyze the biological data using various computational tools. So sequence generation. First generation, we get two techniques, Singer and Maxim Gilbert sequence. And second generation, we get Illumina, PyTorrent, Pyro sequencing, solid sequencing, and third generation sequencing, we get PacBio, Oxford, Nanopore. If we see in picture, read length only expected for the first generation was as 1000 base pair. Okay, and as for the second generation, the read length is expected from 100 to 500 base pair, while in third generation, the read length is more than the both first and second together. That is 10 kb to 100 kb. What are the instruments for the sequencing? First generation sequencing, Sanger sequencing, Maxim and Gilbert sequencing, Sanger chain termination that are used for the short reads, and second generation 454, Solexa, PyTorrent, Illumina. These all produce short reads, so they didn't produce any long read for more accuracy. But third generation produce long read sequencing. We use PacBio, Oxford Nanopore, Illumina, NovaSeq series. If you have any question, you can ask. Anyone have question? So if you don't have any question, thank you for your time. And now I'm stopping my screen. So if you have any question, let me know. Yeah, we will provide recording later.